Why do some people who have a mutation in the GBA gene get Parkinson's disease and others not? We would like to thank our two main funders for this study, the Michael J. Fox Foundation for Parkinson's Research and the Silverstein Foundation for Parkinson's with GBA. We'd also like to thank the Jim and Mary Carnegie Charitable Trust, the McGee Fellowship, the Helen Paul and Ian MacDonald Memorial Summer Studentship, the Health Research Council and Lottery Health Research New Zealand. Without your help, these studies wouldn't happen. So GBA mutations are numerically the most significant genetic risk factor for Parkinson's disease, but not everyone who has a GBA mutation gets Parkinson's. So how is GBA related to Parkinson's disease? Well, the simple fact is that GBA encodes the lysosomal enzyme glucocerebrosidase. When glucocerebrosidase levels drop, alpha-synuclein levels seem to go up, and of course alpha-synuclein forms part of the major deposition of the Lewy bodies that are classically diagnostic of Parkinson's disease. But you can have reduced levels of the GCase without having any mutations inside the GBA gene itself, and you can still develop Parkinson's disease as a result of this. Why is that? Well, we think that basically the mutations inside the GBA gene are not fully penetrant. That is, that if you have them, you don't necessarily have to get disease at all. And that may be because there are modifier genes, genes that are regulated to compensate for changes inside the GBA gene. And in this study, we basically looked for those modifiers. And the way that we looked for the modifiers was quite simple. Our hypothesis was that the GBA gene itself contains switches that control other genes which can modify the onset of Parkinson's disease. And they do that because the regulatory connections that form within the three-dimensional structure of a cell's DNA are important for controlling genes. So a region of a DNA can actually act as both a gene and a switch for another gene. Switches form in cis, that is on the same chromosome, trans-interchromosomal, where they are a long way apart on the same chromosome, or trans-interchromosomally, in which case they occur between a gene and a switch on a different chromosome. And collectively, these switches regulate the genes that are physically involved in the interactions. So we looked for the 3D switches within the GBA gene that might explain why its expression can change without a mutation. And we screened 128 common SNPs within the locus to see what their effects were on genes in cis and trans. Now, 73 of the common SNPs that we checked actually regulate the expression of GBA, and here they are across the locus here. But the key ones that we noted were the ones that were actually within GBA itself. Now, these SNPs, these common SNPs, regulate GBA expression in peripheral tissues, some of which display alpha-synuclein pathology. But importantly, they also regulate other genes, modifier genes, that are expressed in other tissues across the human body. And this, we think, is very important for the onset of Parkinson's disease. 3 of these non-coding SNPs form a haplotype, which is actually associated with a 6-year difference in diagnosis and a 5-year difference in symptom onset age for Parkinson's disease. And you see that clearly in these graphs. Now, the key thing about this is that that BB haplotype actually has a much, much earlier onset than the AA haplotype. And this is really very important because this indicates that these modifier genes are actually having a significant impact on Parkinson's disease. Not only that, but it's also dosage dependent. Now, we also found that there are 6 regions across other chromosomes that co-regulate GBA expression, specifically in the substantia nigra and the cortex. We argue that these regions are maybe responsible, maybe affecting the potential motor and cognitive symptoms that are also associated with Parkinson's disease. Now, the 3D structure of the DNA is different in different organs and different cells. And you see that here we have these different balls, same information but different structures. And so are the switches that form, and the genes they turn on and off. And it's interesting to note that some of these have reciprocal effects on GBA and alpha-2. And so in the brain, GBA is turned on, alpha-2 off, and in the pancreas, GBA is turned off and alpha-2 on. Our project brings a new perspective to the role of non-coding variants in the regulation of GBA and the development of Parkinson's. Common changes in GBA and elsewhere in the genome affect GBA expression, and these changes are associated with the development of Parkinson's disease. And really this emphasises the importance of regulation and gene regulation in Parkinson's. And we argue that the regulatory effects of these common variants that are located within GBA may actually provide a plausible explanation for the way the phenotypes are variable in the development of GBA-centric Parkinson's and Gaucher's disease. To finish up, I'd like to thank William and Sophie, who were the first authors on this paper and did the bulk of the work, Oscar and Martin's team for the nanopore sequencing work, which was really great, and Anthony Cooper, who is a co-senior author with me on this study. Thank you very much. And if you have any questions, please feel free to contact us.